All right, I guess that other one wasn't the last one. This really is going to be the last prokaryotic gene regulation lecture capture, and I swear, this time it really is. Yeah, I know, it is kind of rude to dump this one. But this one is important because it's a global regulatory mechanism to help coordinate the expression of many, many genes. Again, in a prokaryote, we're talking proks here, right? They're all, if the cell's in an environment, it's in an environment. If something changes, the cell needs to change. Whereas in a multicellular organism, there could be an environment in one cell that has nothing to do with the environment of another cell. So they're more individual. So if there's specific situations that prokaryotes need a whole set of genes, not just from an operon, but a whole different set of lots of genes, it would be great, and it would be, I don't want to say smart, but it would be evolutionarily advantageous to be able to deal with that. And so guess what? They can. And what we see, what we were talking about in class was under normal temperatures, normal situations, no stress, nothing crazy going on, the bacteria, the normal sigma factor, which is actually sigma 70, that's its real name, right, binds to RNA polymerase and recognizes the sequence in the promoters to initiate transcription, that -10 and the -35 site. Right? Just like we said. So I didn't lie, right? I know, shocking. If they're at high temperature, which we'd call a heat shock, so like let's just say, for example, you are strep throat, a streptococcus aureus in somebody's throat. If that you have a fever, your temperature is elevated, it's not normal anymore. The elevated temperature can disable normal sigma factor. What does that mean? It doesn't work anymore. It can't do its job. At high temperatures, what can work? Ooh, a different sigma factor becomes functional, right? Conformational change. This one kind of probably falls apart. This one's conformation becomes able to bind to RNA polymerase and to a -10 and -35, but a little bit different sequence. Not the same one as that, but a consensus sequence at both of these sites, okay? These would be genes that help the bacteria survive at higher temperatures. These are the normal sequence at -35 and -10. Sigma 32 would be the heat shock, right? When we raise the temperatures, just like I said, the sequence is at, well, close enough, -39, and at -15, so very close to -10 and -35, recognizes this sequence. So any promoters that have this would be turned on by sigma 32, only active at higher temperatures. So if the temperature is normal, we're only going to get genes turned on that have this sequence in their promoters. Higher temperatures, genes turn on with this sequence in their promoters and not this one. If you happen to be infected with a bacteria, a virus, a phage, a bacteriophage, then a different sigma factor is activated that recognizes just one single sequence at the -15. So any promoters that have this sequence at -15 would be turned on when they're virally infected. So they could have this sequence and these, and then be turned on all these genes, right, that have this one. There'd be some overlap. And if you look, here's -15 and -15, so these guys are going to be turned on. Whatever has this is also going to be turned on by sigma 23. So it's just another way to turn on a whole huge set. Anything that has this under normal is going to be on unless the repressor proteins are bound, right? You're still going to have that kind of activity, but it's possible for them to go. If it's heat if it's at higher temperatures, only these guys are going to be possible to go. And then they'll be regulated by protein DNA interactions too, likely. And again, anybody with this, right, has the opportunity to be turned on when it's virally infected. So this is just an example, right? The sigma 70, right, minus 35 and minus 10, sigma 32 would recognize this type of sequence. So that's the way to coordinate expression of many genes. And that's it. I said it'd be short, and we're done. Thanks for listening.